I'm Sunita Chandrasekaran, an associate professor at the University of Delaware. And with me, I have three wonderful co-authors, and each of them have a story there. So the first one is Matt Stack, who was a primary developer for this particular project, which is the OpenACC acceleration of this agent-based biological simulation framework. Matt Stack was not in NVIDIA when he was the primary developer of this project. He was a rising undergraduate senior researcher in my group, graduated in 2020, and then joined NVIDIA. So hats off to Matt for carrying on this work while he was graduating with his undergraduate degree. Then is Paul Macklin from Indiana University, he's a professor there, and he is a system biologist, computational scientist, mathematician, so wears lots of hats. And I heard him talk about this work, the PhysiCell biological simulation framework, in a cancer workshop. And I noticed he was talking about how models and simulations were running for a weekend and more. So we're looking at 72 hours. So that got me approach him later on, and we were sitting by, at a bar, and we were scribbling at the back of a napkin and wondering, so can we make this go faster? Because then we could do a lot more simulations faster, not just one simulation, and reduce the time taken. So that's how we started collaborating, and it's been a fun collaboration. Robbie Searles has been a mentor on this project. I also graduated from my group in 2019 with a PhD and wears his NVIDIA hat on. This paper is under review, so there is a preprint link. Also, the OpenACC code of this particular project is open source, and the PhysiCell project itself, the main code, is also open source. So the entire project is open source. Feel free to take a look. Stepping through, oops, what just happened? Okay, stepping through. So a little background. Immediately, I'm sure you're all looking at the right-hand side video there. There's lots of stuff happening, lots of cool stuff happening. So you see a lot of cells. And the project is all about, how different cells interact, different types of cells, different properties of cells involved, and how does, say, an immune cell correspond with a tumor cell, and then what happens to the different biological transport of many diffusing factors and so on. So human body is complex, and add a tumor to it, things become even more complicated. And now let's think about, how do we program this, right? How do we create a code? How do we create data structures, templates, and make this a modularized code? So that is what is the background of this scientific, this science. It's called PhysiCell. So the PhysiCell has been working on workstations, and obviously the goal is to move this on to larger clusters, HPC clusters, MPI, OpenMP, OpenACC, and all the jazz. The work also has been adopted in cancer and infectious diseases and complex biological systems. So again, a little bit more motivation as to why this got us really excited. So what you see on the right-hand side is a beautiful visualization video, right? And these are the perks of working with non-computer scientists, because you get these cool videos and you get to demonstrate how you can apply computer science on a real-world problem. So what you're seeing is yellow cells, blue cells, which are tumor cells. Yellow cells are aggressive, blue cells are less aggressive. And you also see some black cells, which are consuming oxygen, and they would die in case of low oxygen. Now, the same thing is being mapped onto a time scale. So you see a tumor is starting off at 0 days, and it's at 7 days, the tumor is on, and you're adding some immune cells to it, which is the red cells around the tumor cells. The immune cells start interacting with the tumor cells. They get in contact, and they do what they're supposed to do, and they start killing the cancer cells, tumor cells. However, immune cells escape, right? So the tumor cells can grow back, which is why on the 21st day stamp, you still see a lot more yellow cells. Those are the aggressive, proliferating rapidly cells. So this is a 7 to 14 to 21 day simulation. And you notice that this is taking about 1 to 3 days, which is 72 hours of computation on a desktop workstation. And this is a problem, because you want this to happen way faster, right? To be able to create clinical therapeutic medications for this. And so you cannot afford to stare at this for 21 for 3 days altogether. So that motivated us to take on this challenge. And our goal here is to accelerate the agent-based biological simulation framework. And obviously, the overall goal is to reduce the time taken to be able to explore newer biological dynamics. And we also want to maintain a single source code because not everybody has access to GPUs, right? That they might have access to multi-core systems. So they should be able to take the code, run it on the multi-core without needing to change any source code at all. So that is another goal. And we want to be able to allow 3D multi-scale simulations for cancer and diseases and advanced cancer patient digital twin. So as many speakers have already talked about earlier, you start off such a massive code base with profiling. And I listed some of the common profilers we have been using in our team. And I guess most of you are familiar with almost all of it here. So that's what we did as well. Identify in this complicated C++ code base, where do we start, right? So that was the first bullet. And where did we start? So what you're seeing is this little ring, which has a rusty orange, a teal. Those two are, dominating the ring. What you're seeing is the rusty orange color is the biotransport or the diffusion part of the code. And this talk is focusing on how we accelerated that portion of the code. So we haven't yet accelerated the entire code base. So we started small. The teal is where the cell updates are happening. And there are little other portions in the ring, which is saving data, initialization, etc. The key takeaway here is you're seeing a number of diffusion steps, 10 diffusion steps for every single mechanic time step, and 600 diffusion steps for every single cell time step. So you can notice that the diffusion steps are dominating, right? So that's a 66% diffusion and biotransport, and the rest are 30% and so on and so forth. And thanks, Paul. So he was mapping this on the scale in the bottom, and that's exactly what you see. So we focused on the diffusion time step and started applying OpenACC programming model. So as you can imagine, there are lots of code cleanup, right? And at the same time, we didn't want to mess with the algorithm because we need to preserve the algorithm to be able to verify that our GPU code and CPU code are accurate, right? So we need to be able to do that. To that end, we managed to make sure that we didn't change the algorithm too much for this particular application. So the logic, the cell logic stayed on multicore, and the diffusion, the low-level mechanics and the cell-to-cell mechanic interactions that you saw on those visualization videos that you saw, all that, the goal was to move this to the GPU. With that as our goal, diffusion step, the rusty orange thing that you saw in the ring, entails a number of computational steps and function calls. So how did we go about that? We started copying data. The speed density vector, which deals with the cell secretion, forms the main data on GPU. So we started copying the data that was really essential into GPU, started initializing them. Whenever there was an update required, we either used user-managed memory or OpenACC managed memory, and we will show you results of how they fared either way. Then one of the challenges, or one of the most important challenge we faced, and Matt spent a lot of time working around this, was that data in its original state on the host was in vector. We needed to translate them to arrays to be used on the GPU. And here we are dealing with, the speed density vector itself is a pointer to a standard vector. So it was quite complicated to translate everything to arrays. You need to make sure you're not introducing bugs, which we are very good at. So that took a lot of careful analysis. And we also used this keyword, thanks to OpenACC, for this particular feature, because this was important for us to let the compiler know that we need to keep the newly created data and all the surrounding data on the GPU. So a wish list here, I think this has come up often, and there might be reasons as to why this is a challenge to support on the GPUs, but standard vector support on GPUs would have been useful for us. Then another thing that we would have found useful is adding a runtime command tool like the acc_notify to understand, how much of, how much memory and in what direction they are headed, between host to device and device to host. So some sort of tooling functionalities would have been useful for us to step through some of the translations that we had to do. That's a little code snippet, basically summarizing what I just mentioned about this keyword, a little deep copy there going on, but that forms the main structure of the p-density vector, which forms the main data component of the diffusion step itself. Then other types of OpenACC applications we did was move the blast type function, which has been inlined in each of the XYZ diffusion functions on the host, but to make it more readable and make it more clean, we replaced that with a single diffusion function on the GPU. It improved readability. And as you can imagine, lots of conversations went back and forth with Paul about several other functions, including the Dirichlet condition, which we thought could remain on the host until we decided that it would rather be on the GPU because it eliminates a lot of high rate of updates with fresh data between the Dirichlet and the diffusion computation steps. Then we also used ACC routine to wrap up some of functions for GPU execution. And that's a laundry list of functions that made to GPU eventually. The first two are the blast type functions. The transfer 2D and 3D were those functions that dealt with data for the diffusion steps, but not necessarily on the GPU. They remained on the host for the most part. Then translate vector to array, array to vector were those important functions that took care of translation between vector to array types so that we could port those data on GPU. Then the Dirichlet function, and then the diffusion 2D and 3D, those 5 were the main diffusion computational steps for both 2D and 3D functionalities, and the sizes of the p-density vector. This is a quick snapshot of nSight system, where you can see on the left-hand panel the different portions of the GPU. The key thing to note here is the green bars that you see at the initial steps. Those were the memory transfer to device, but you also see lots of blue blocks, which is great because you don't see a lot of movement between host and device. So less data transfer is good. So you see a big blue block, which was great, which means lots of homework was done to make sure that we kept the data transfer in the beginning and only take care of computation towards the latter half of the computation. Now comes the fun part, which is the experimental results. And we used DGX2, A100, and V100, stepping through. On the left-hand side, you see the different computations we did. 60 simulation minutes is one case study, and I'll show you 2 more case studies in the next slide. So what you see here is CPU 1 core, OpenMP CPU 1 core. So you must be wondering, is that the serial code? No, it was the parallel version running serially, so single core, because that's what we had access to. So you look at that, and then let's look at the last row, which was the managed memory on A100. So between 1 core and A100, 524 seconds to 21 seconds, we see about 25x speedup. But if you were to compare multi-core versus and managed A100, you're looking at about 2.72x. You also see time consumption by the user manually managed memory implementations, and they're not doing as well as the managed memory did. Now, this was using NVC compiler 21.3 and with the input set representing a cell size of 1 centimeter. Now let's propagate this to 180 and 360, right? And the bottom line here is when it was a 360 simulation minute model, which means roughly 5 hours, we're looking at 82 seconds on the A100, which is really 37x speed up compared to the single core run, or about 4x compared to the multi-core run. Now, we need to think beyond this, right? So this is a 5-hour 360 simulation minutes, and that's how quickly it was running as opposed to a multi-core or a single run. Now think about a 21-day simulation, right? So how much more you can speed up and how much time you would save if you had this accelerated code. I showed you raw numbers. So what you're seeing is the speed up. There's no new information here, but it's basically speed up over normalized over serial. And you clearly see as to how the GPU bars stood tall. I have just 2 more slides. So this one is basically communicating how The 21-day simulation, an estimation basically, if you had a long time simulation, not just 360 minutes, you're looking at about an hour, 42 minutes compared to CPU only for 9 hours. Now think about clusters, right? You have a cap as to how many hours you are allocated. So if you only had 4 hours, and if you had a 21-day simulation, and you're going to take 9 hours on the CPU only, you won't be able to complete the simulation, obviously. Rather, if you have an accelerated version, you're able to do that so quickly. And you can also do more than 21 days in that 4-hour timestamp, say 42 days or even more. So that's where we want to head to. Quickly, we also verified and using different methods to make sure that the CPU and the GPU results were correct and accurate. Depending on 1D and 3D simulation, we had different types of verifications that we were able to do. And we also looked at the bitwise identical results to make sure that the 3D simulation was correct. Sometimes we also used visual inspection. You clearly see before and after, right? There was a bug in the Dirichlet condition. This looks funky, but upon fixing visually inspection of the code, you see that this is definitely looking way better. So to conclude, cutting edge science requires about hundreds to thousands of long simulations, right? So it's not just 360 simulated minutes. And long simulations would enable dynamics that only emerge at longer times. You wouldn't be seeing that at 60-minute simulation anyway. Forecasting disease progression over weeks in time. And the many long simulations can also enable large-scale model exploration, data assimilation for individual patient disease forecasting, digital twins. It can simulate better the cone of uncertainty and so on. So optimizing the diffusion step, the rusty orange ring, the biotransport reduced simulation time by 50%. That's 2x improvement in scientific throughput. Significant advance towards those 100,000 of long simulations, which we are yet to do. And another estimate is optimization priority would be 10 GPU steps per CPU step to 600 GPU steps per CPU step could easily give us 4x. And that's what we want to do, because the problem is more than, just running the shorter timestamp simulations. Thank you.